The aim of the Tumor Target Screener Appyter is to compare the gene expression patterns in a patient tumor with gene expression patterns in normal tissues and cell types. The goal is to identify genes that are only highly expressed in the tumor while lowly expressed in all other tissues. A link to this tool can be found in the video description. To accomplish this, we first process and organize data about gene and protein expression in normal tissues and cell types from multiple sources. These sources include the GTEx transcriptomics and proteomics datasets, transcriptomics data from ARCHES-4, which provides uniformly aligned RNA-seq data from the gene expression omnibus for over 1 million samples, Tabula Sapiens, which has gene expression data from over 450,000 single cells covering over 460 human cell types, and proteomics data from the Human Proteome Atlas and the Human Proteome Map. The Appyter takes as input tumor expression data and ranks significantly differentially expressed genes across all tissues and cell types. Tumor sample data can be uploaded from the user's local computer or via URI pass-through using one of the supported protocols. If the user has an Appyter account, files which were previously uploaded to Appyters or files from any integrations, such as CAVATICA, can also be used. By default, the Appyter will run using public Appyter resources. After uploading the tumor sample's data, the user can select the normal tissue background to compare to. There is also support for backgrounds at the transcript level, which can be used to identify differentially expressed isoforms. The user also has the option to restrict the target list to membrane proteins. This is done because plasma membrane proteins can be used more readily as targets and biomarkers. Their location on the surface of cells makes these proteins accessible to antibodies that can be developed to specifically target them. The first step of the Appyter is to align the distribution of the expression data from the input to the background. A Venn diagram is shown with the overlap of gene IDs between the input and the background. The next step involves computing the differentially expressed genes. The results are displayed as a volcano plot with the significantly highly expressed genes in the tumor colored in red and blue. Blue points denote membrane proteins. The top 10 most significantly differentially expressed targets are listed in a table, and the results are visualized in a series of box plots that compare the expression of each identified gene target in the tumor to the expression of the gene across all other tissues and cell types. These visualizations can be used to identify the unique normal tissues and cell types that may be impacted by targeting the gene that is highly and uniquely expressed in the tumor, for example, with an antibody-drug conjugate. One important aspect of the Appyter is that it can run within the CAVATICA patient-protected environment using CAVATICA resources. This enables the analysis of any protected health information, or PHI datasets, as well as very large datasets using high-end computational resources. CAVATICA integration also enables the Appyter to easily become part of other workflows already implemented within CAVATICA. To accomplish this, users will need to set up accounts in both Appyters and CAVATICA. Appyter accounts may be set up through OrcID, GitHub, or email, while CAVATICA supports both email and ERA Commons logins. Once such accounts have been established, instructions on how to connect these accounts and execute Appyters within CAVATICA are provided in the Appyter user account documentation under the Integrations tab. The user can then return to the Tumor Target Screener Appyter page and upload data from a CAVATICA project, then choose to run the Appyter using CAVATICA resources.